This is part two, excuse me, this is part two of the chapter one lecture notes on viruses. We're starting on slide 8 with viral receptors. So you're looking at a virus, and actually you're looking at the attachment step where the virus attaches to the host cell. And it says in the notes, many viruses use some sort of glycoprotein to attach to their host cells via molecules on the cell called viral receptors in order to penetrate the cell membrane. So um, the okay, and just to make that, I was just reading that, and it's kind of confusing how it's written. But there is a receptor on the host cell, as you can see here, called the XCT receptor. And that is a molecule that is located within the phospholipid bilayer, the plasma membrane of the host cell. You can tell this is a eukaryotic cell. And there is also a molecule that is called the viral receptor, which is located within this particular virion, has an envelope. This is the capsid, which has a hexagonal structure. And then this is the nucleic acid core. But then this outside portion is the envelope. So it's within the envelope that the viral receptor is found. And it's the viral receptor that attaches to the host cell receptor in order to penetrate the host cell. This virus is called a bacteriophage, and we actually did learn about bacteriophages. This is bacteriophage T4, but bacteriophage is a virus that specifically infects bacteria. And this bacteriophage T4, is an example of a virus with a complex shape, complex structure. This particular one has a head region. The DNA is located within the head region, and then it has a tail region with tail fibers. I was calling them legs or appendages, but it's the tail fibers that actually attach it to the host cell, and the host cell is some sort of bacterium. Another shape for viruses is this shape that you see here that's seen in an adenovirus. This one does not have an envelope. It simply has a capsid. Looks sort of spherical, composed of capsomeres, and then the glycoproteins are attached, are located within the capsid. The DNA core is inside the capsid, and those glycoproteins, remember, are used for attachment to the host cell, but there is no envelope. This is a illustration of an adenovirus. Adenoviruses are transmitted orally. They can cause eye infections and respiratory infections. They are double-stranded DNA viruses. And the shape of their capsid is not spherical. It's actually called icosahedral. So that shape, that geometrical shape of that capsid, is called icosahedral. And HIV, of course, human immunodeficiency virus, which causes AIDS, acquired immune deficiency syndrome, is a retrovirus. And its structure is what we're studying here. Retrovirus means that it has RNA, which is converted to DNA with the enzyme reverse transcriptase. And then the DNA goes through transcription and translation. And we learned that RNA viruses and retroviruses in particular tend to undergo mutations at a very fast rate. So it's hard to make a vaccine. It's almost impossible to make a vaccine for them. But you can see, as far as the structure of this virus, this is the capsid, composed of capsomeres. Here is your RNA, which is kind of in segments here, but there's your RNA, and then these little enzymes, they're kind of orange, are the reverse transcriptase, which converts the RNA into DNA. And then HIV has got an envelope outside of the capsid, and the glycoproteins are located within the envelope. There are two viruses shown here, the rabies virus and the variola virus. The variola virus, what you're seeing in this image in picture B, is someone with smallpox. And the reason that you've probably never seen this before, unless you've seen it in a photograph, is because worldwide we have eradicated smallpox. We have a vaccine for smallpox that children get, and it is extremely rare. You almost never see anyone with smallpox. But when smallpox, before the vaccines became available, when it did infect people, it if they lived through it, it left pock marks. You may have heard that someone's skin is pock marked. And that's the marks that are left in the skin, the scars that are left from the smallpox. Okay, so viruses can be classified by genetic material and capsid design. And here what we can see is you have the rabies virus, which is has got a capsid, and then it's got an envelope outside, and it's got single-stranded RNA for the genome. So it's got a capsid. It also has an envelope. It's an enveloped virus containing single-stranded RNA, and that's how it's classified. And then the variola virus, which causes smallpox, also contains an envelope, a capsid, but its genetic material is double-stranded DNA. So that's how it's classified. It contains an envelope and it contains double-stranded DNA. This chart gives you some examples of viruses classified by their genetic material, by their core. So some RNA viruses are rabies and retroviruses. Some that are not listed on here that are RNA viruses are the cold virus, HIV, which we know is a retrovirus, the flu virus. And DNA viruses, some examples are herpes viruses and the smallpox virus, which is variola. Okay. You can see that it also classifies rabies and retroviruses as single-stranded, so they are single-stranded RNA viruses, and the herpes and smallpox virus are double-stranded DNA viruses. Then the chromosome, which is composed of the DNA, can either be linear or circular, or it can be a little bit more complicated than that. So the rabies, retrovirus, herpes virus, and smallpox virus all have linear chromosomes. So all these that we looked at up here have linear chromosomes like humans do. And then the papillomaviruses, that includes a virus called HPV. You may have heard of this because they have a vaccine for this out now, that they encourage children at around the age of 11 to get this vaccine. HPV is human papillomavirus, and it causes genital warts. And when a person gets genital warts, that can lead later on in life to cervical cancer. So they try to encourage children to get that vaccine because it can prevent cervical cancer in females. And they encourage the little boys to get the vaccine, of course, because then they can't transmit it to a girl, So papillomaviruses like HPV, many bacteriophages, which of course only infect bacteria, will have circular genomes, circular DNA, or circular chromosome. Parainfluenza viruses contain a genome that has a single segment of genetic material, and then influenza viruses, which cause, flu, contain segmented. That means the genome is divided into multiple segments. So then we have classification by capsid structure. There's no need for you to memorize this information, by the way. I know I'm throwing a lot of information at you. There is one classification system, and I'll go ahead and show it to you, the Baltimore classification. this is the one that I tend to focus on more, as far as if I was going to ask a question on an exam. The Baltimore classification is the best one I think that's out at this point in time. So let's go back. Okay, so in this classification, we're looking at the capsid and some viruses that have naked icosahedral capsids. That means they don't have an envelope. The word naked means they don't have an envelope. Are hepatitis A and the polioviruses. Some enveloped icosahedral are Epstein-Barr virus, herpes simplex virus, rubella virus, yellow fever virus, and HIV-1. And some enveloped helical are, it means the capsid is a helical shape, are influenza, mumps, measles, rabies. And then naked helical, you saw that was the very first image that we looked at of a virus. Is the tobacco mosaic virus, which infects different types of plants, including tobacco. And then complex capsid structure, like the structure of the bacteriophage that we learned about, the T4 bacteriophage, that was a complex structure. It had a head and a tail, and the tail had those attachment proteins, that look like little legs. But anyway, herpes virus, the smallpox, variola virus, and hepatitis B, and the bacteriophage has a complex capsid structure. And here's some images. Picture A is naked icosahedral. Picture B is enveloped icosahedral. Picture C is enveloped helical. And picture D is naked helical. You may recognize this one as the tobacco mosaic virus. And then picture E is complex. These are all images. They're all images taken with electron microscopes, just so you can see some different. The main idea is that viruses have different shapes. I mean, it's really, really simple. We're just showing you some of those different shapes. Okay, so now in the Baltimore classification system, I do want you to focus on this one when you're studying viral classification. The Baltimore classification uses the genetic material of the virus as well as, it focuses, I'll just say that it focuses on the genetic material and the way that messenger RNA is produced. So it's focusing on transcription. So we have 7 groups of viruses if you use this classification system. This classification system does not focus on the capsid or the envelope. It just simply focuses on the genetic material and the way that messenger RNA is actually produced. So we have group 1, which has double-stranded DNA. Easy to understand because this is identical to what we learned in Bio 111. The DNA is transcribed into messenger RNA, and the messenger RNA is translated into protein. So herpes simplex viruses, the herpes viruses, are examples of group 1. Then you can have single-stranded DNA. And in this case, it still keeps it pretty simple because your single-stranded DNA simply converts itself into a double-stranded form and then transcribes messenger RNA, and then the messenger RNA is translated into protein. So it's just one step extra. Parvo, you may have had a pet that had parvo. It's a very dangerous virus, causes diarrhea and vomiting, and it basically causes the animal to, dehydrate, and usually they die. But they can. I've had a pet with parvo. It's really sad, but they can be saved sometimes if they get to a vet and they get them on an IV, or if you just get enough like Gatorade or some type of fluid with electrolytes in it. Anyway, it looks like our time is running out, so I'll come back to this in the next lecture.